Welcome back, everyone. In this lecture, we're going to talk about images and NumPy. NumPy by itself can't actually directly open a .jpeg or .png extension file. Instead, we need to use it in conjunction with what's known as the PILO or PIL library, which stands for Python Imaging Library. Let's go ahead and open up a notebook and take a look at how this all works. Alright, so to begin, we're going to start by importing NumPy as NP. And then we'll also say import matplotlib.pyplot as plt. Matplotlib is a library that's actually going to allow us to plot out and display images inside of the notebook. Now, in order to display things inside of the notebook, we also need to write out %, matplotlib inline. Then go ahead and run that. Keep in mind, as a quick note, the actual latest versions of Jupyter and Jupyter Lab don't require this matplotlib inline, but I'm leaving it here just in case because it doesn't do any harm. And sometimes there's bugs if you don't have that actual line. So that allows us to display plots inline in the actual notebook. Now it's time to actually try to open up an image with NumPy. Now in order to do that, we first need to open it using the Python imaging library. So we'll say from PIL in all caps, import image. And notice I is capitalized there. So this is an image function that allows us to open up images, and actually then we're going to be able to transform this image into an array that NumPy can understand. So I will say pic is equal to image.open, and then you can just provide the file path for any image. Now if you downloaded the course notes, you'll notice that there's a data folder here, and there's a file called 00-puppy.jpg in the data folder of our actual notes. So that's what I'm going to provide the file path there. So I will say data 00-puppy.jpg. You need to make sure that you actually are providing the right path. So in my case, this data folder is actually in the same location as this notebook, but if you're one location deeper, you may need to say .. So it really all depends on where this notebook's actually located in conjunction to your actual JPEG file that you're trying to open. If you are unsure, just go ahead and pass in the whole file path, which is going to be something like C, users, then your username, and so on. So just keep that in mind, otherwise you'll get an error. So I'm going to now collapse this. So looks like I got the error right now. I'm going to undo that /. There we go. So now I'm ready to go. And if I just take a look at this pic and do Shift Enter, I get back this pic of this adorable puppy. Now let's go ahead and take a look at what type of object this is. So if I were to say type pic, it's a right now Python imaging library or PIL JPEG image plugin. It's a JPEG image file. So right now it's a JPEG, which means that NumPy can't actually deal with it. But what we can do is transform it into an array. So we're going to say pic underscore ARR is equal to np dot as array, and then pass in that pic. And now if I take a look at the type of pic array, it's now a NumPy array. And in fact, let's take a look at its shape. So its shape right now is 1300 by 1950 pixels with 3 color channels. And if I say plt.imshow, which is going to allow us to show this array as an image, I'll pass in pic array, run that, and then I get to see the actual array laid out. So here's the image, and this plt.imshow, it's kind of a special function that's specially designed to show images that are transformed to as arrays. Now notice that we get these grids. So I can see here how it's 1300 pixels in length and about 1950 in width. And there's 3 channels here, the red, the green, and the blue. So let's actually access one of these channels by itself so we can see what's going on here. So I'm going to say pic red is equal to pic ARR, and I'm going to make a copy of it. So previously in the NumPy lecture we showed you how we can make copies. Now if I actually just display this red image, I get back right now the same image as I did before, which makes sense because it was just a copy. However, let's try the following. We're going to try to zero out the contribution from the green and the blue channels. So what we're going to do is if we're to take a look at this array, it's 3 channels right now. One 1300 by 1950 array for red, one 1300 by 1950 for blue, and another one for green. In our case, it's going to be RGB, or red, green, blue. So how do we actually zero out these values? Well, what I'm going to do is say pick red, and remember the shape of this is 1300 by 1950, essentially 3 arrays of 1300 by 1950. I'm going to say pick red, and then I'm going to grab all the rows and all the columns in the green channel. So the way this is formatted is it goes red channel, then green channel, and then blue channel. So each of these shapes, one stands for red, one stands for green, and one stands for blue. So if I take a look at just this, which means everything in that 1300, everything in 1950, but just one, that actually stands for the green channel, because R would be 0. So let's go ahead and display this. We'll say plt imshow, and you'll notice something funny happen here. You'll notice now the coloring seems very strange, and that's because if we take a look at these values, they're now all between 0 and 255, and Matplotlib by default will show this using what's known as a Viridis color scale. So when we ran imshow on this, so if I say plt.imshow, it took those values and applied some sort of color scale. And the default, like I said, is Viridis. If you actually check out the Matplotlib documentation on their color codes, they have perceptually uniform sequential color mappings, and Viridis is this one that goes from purple all the way to yellow. And it's actually specially designed for people who are colorblind, and these are all actually designed for people that are colorblind. However, this actually isn't really an accurate representation of what this particular array represents. In fact, what's probably more accurate is if we were to say cmap gray and then run this. This stands for color mapping, and we choose gray, which essentially just says show this in grayscale. So this right now that we're showing right here, this is the red channel values. They're all between 0 and 255. Remember, there's 2 more of these, one for green and one for blue. So let's check it out. This is the one for green, and then let's check out the one for blue. You'll notice that they're all a little slightly different. So for example, if we take a closer look at the red channel, remember 255, that means a full pure red. 0 will then mean no red, pure black. So that means when we're actually viewing this in grayscale, the closer to white, that means more red for this particular image. So if we take a look at the ears here, they tend to be pretty white, which means there's high values of red there. And if we were to scroll back and take a look at the actual dog's ears, you'll notice that his ears actually do contain quite a bit of red. It's kind of an orange color. But if we take a look at the other colors, such as the color mapping for green or the color mapping for blue, you'll notice his ears appear now quite dark. That means there's not a whole lot of green in his ears. There's a little bit, but there's definitely not a whole lot of blue. So again, here we're displaying the 3 different channels as grayscale. So this is red, because it's the first one we indexed at 0. This is green, the second one we indexed at 1. And this is blue, we indexed at 2. So again, take a look back at the slides of what is an image to better understand what these 3 images are actually showing you, because it's kind of hard to understand without the full context of the what is an image lecture. Okay, now back to the task at hand. Let's imagine that I actually made all these values 0, which means I said the following. I'm going to grab my pick red, and I'm going to set everything in the green channel. If we just take a look at this, we have all these values from 0 to 255. I'm going to set these all equal to 0. I'm going to 0 them out. So now I've completely gotten rid of the green channel. So let's see what our picture now looks like. We'll say plt imshow pick red, and you'll now notice that it looks a lot stranger. In fact, it looks a lot more purplish, and that's because the only two colors that are left are blue and red, which together make purple. So now we're basically seeing everything without the color green. Now let's take this a step further and remove the contribution from blue. So what we're going to do here again is say pick red, and I'm going to grab all the pixels, except now I'm going to grab them in the index 2 channel, which is the blue channel, because it goes red, green, blue, and we're going to set that to 0. And now when I see pick red again, I've completely gotten rid of blue. Essentially now there's then no more purple, and we're seeing the red channel completely. Something to keep in mind is this pick red, its shape is still 3 channels across. So a lot of times students are a bit confused as to what's the difference between pick red here versus the pick red that we were seeing earlier when we actually just grabbed one. And that's because we're seeing all 3 channels at once instead of just one channel. So if I were to take the shape of this guy, this is just 1300 by 1950. So this is just one single channel. And if I do the same for the other ones, you'll notice the shape doesn't change. When I say pick red without any indexing, it still has information for all 3 channels, except cleverly, we went ahead and zeroed out the green channel as well as the blue channel, which is why this picture only shows up with the red. Okay, so I know that was a bit complicated because of the NumPy indexing involved, but the actual idea is quite simple. Essentially, we know the image has 3 channels, a red one, a green one, and a blue one. We changed all the values in the green channel and the blue channel, those 2 layers, we made them all 0, which means we basically told the image, hey, there's no longer any contribution from green and also no longer any contribution from blue. So then when the image or PLT was asked, hey, can you show me what this adjusted image looks like? It could only show what was in the existing red channel, which is why everything's now colored red. That's different than what we did before, which was color mapping a single channel to grayscale. Okay, if you have any questions, feel free to post to the Q&A forums, but definitely do another watching of the What is an Image to further understand what's going on here. Thanks, and we'll see you at the next lecture.